Hi, so welcome to the May session of MycoTalks. So today we're going to have two speakers as usual. So beginning with Georgios Chamilos from Greece and molecular mechanisms regulating fungal phagosome biogenesis. And then afterwards, Anuradha Chowdhary from the University of Delhi and India, who will be telling us about Candida auris. So if I could just remind you the way that MycoTalks operates. So the philosophy is the program is to have talks once a month on the last Thursday of the month from people at different career stages, working in different fields from different areas. And we'll have two 30-minute talks from our speakers, followed by 30 minutes of in-depth discussion. So can I remind you that we'll hear the first talk and then we'll hear the second talk. And then after that, you'll have the opportunity to ask questions. You can submit questions as the talks are ongoing, and that's actually welcome if you do so. But please remember to put the initials of the speaker before your question so that we know which speaker they're addressed to. And you submit the questions through the Q&A, not through the chat facility. So it's the moderators will see the Q&A. And I'd also like to remind you that recordings of these talks and all the other talks will be available on the MRC YouTube channel. So as well as today's session, obviously we have sessions every month. So coming up in June, the end of June, Agostinho Carvalho from the University of Minho in Portugal and Jaron Lin from the University of Georgia. And then in July, we'll be hearing from Darius Armstrong James and Aaron Mitchell. As well as the MycoTalks, particularly for the clinicians who have Myco clinics. And the next Myco clinic session is scheduled for the 8th of June. Paul Vervey and Neil Clancy will be talking about the very important topic of clinical challenges of antifungal resistance. For the trainees, the medical mycology trainee seminar series would very much like to welcome students, postdocs, all clinical trainees, any new faculty. And it gives you the option to present your work, usually during the second Thursday of the month, though they do vary it. So the organizing committee would very much like to hear from trainees, and you can see the website there below. So it's bit.ly and MMTSS. So if you're interested in presenting there, please do so. Okay, so the session today will be chaired by me, hopefully. So we just have one chair today. So we hope that everything is going to go well. So our first speaker, we're delighted to welcome Georgios Chamilos, who's the Professor of Clinical Microbiology at the University of Crete, and also based with a position at the Institute of Molecular Biology and Biotechnology in Heraklion. So he received his MD from the University of Athens, working later in the University of Houston, and has been back in Crete since 2009. His interests include understanding host effector mechanisms regulating inhibition of Aspergillus fumigatus, pathogenic mechanisms employed by the fungal cell wall molecules that target phagosome biogenesis, and molecular mechanisms of immunoparalysis of phagocytes induced by sepsis and other immunosuppressive conditions. So we're very much looking forward to hearing from you, Georgios, so you can share your slides now and start when you're ready.
Okay. Thanks for kind words and thanks for the invitation. And for the sake of time, I'll go directly to the topic, which is the role of phagosome biogenesis in fungal diseases and how molecular pathways are regulating this process physiologically and what happens and how the defects on these pathways contribute to invasive fungal infections. I don't think I have to explain a lot about the importance of invasive Aspergillosum, mucormycosis, and other fungal diseases. Just to point out that from this cartoon, you can appreciate that although these are considered extracellular pathogens because the invasive form includes tissue invasion by filamentous forms of the fungal conidia, there is an important role of intracellular interaction of the fungal spores, which are inhaled with the innate immune cells in the lung, especially the alveolar macrophages. And this is the focus of our research the last 10 years. So normally, when we inhale conidia, they are rapidly phagocytosed very efficiently by alveolar macrophages, which are sitting on the alveoli. And through mechanisms that are not very well understood, this conidia are efficiently eliminated with minimal inflammation. So in an expanding group of patients with immunometabolic defects, which are not very well understood at the molecular level, this intracellular conidia, which can persist for many hours inside or even days inside the macrophages, they can germinate from this polarized growth of the fungus, which then invade the lung parenchyma, causing catastrophic necrotic infections that you all know as invasive fungal disease, and especially invasive mold infections. And you all know that nowadays we are shifting the epidemiology of these infections from the era of neutropenia, of leukemia and transplantations that were getting these nasty necrotizing pneumonias, to the era of biological therapies, sepsis, new immunomodulators, which are associated, some of them are associated with emergence of invasive aspergillosis and lately mucormycosis, especially in the COVID area. And for this part of non-neutropenic patients, there is not much known about the underlying pathogenesis and the role of macrophage defects on this process. And this is basically one of the interests of our lab. However, we don't take the traditional translational approach to go from the disease patient and understand the mechanism, simply because there is a lot of complexity there. So we follow a reverse pathway. We go through the physiological immune response, and we use animal models from Drosophila to mice and to primary human cells to understand what is the biology of interaction between the fungus from the one side and the innate immune cell, mainly the macrophage from the other, and how these two compete and physiologically, and then in the disease setting, whether novel mechanisms that regulate this interaction for the benefit of the host fail, and this results to the disease. And obviously, our lab has direction towards identifying novel biomarkers to stratify immunodeficiency nowadays. Immunodeficiency is something that is easy to say as a word, difficult to quantify in terms of quantitative biomarkers. And obviously, to understand the molecular defects and harness the host response in a way that we can reverse the underlying immunodeficiency and ultimately to improve disease outcome. And here are some of the strategies and the models that we use from Drosophila, which has a very conserved, especially cellular, but also humoral immune system, to the mouse models and mouse macrophages, to the human macrophages in the physiological people and also in people that have the disease. And our research is centered on the interaction inside the phagosome of macrophages following phagocytosis of conidia. As you all appreciate, the first cells to encounter these spores of molds like Aspergillus and Mucorales are the alveolar macrophages, which interact with epithelia and efficiently eliminate this conidia inside the phagosome. However, if something goes wrong, then we have invasive disease. Neutrophils are coming at second line of course defense, and if this fails also, then you get invasive disease. And for many years, until we started working with the phagosome, not much was known about the molecular pathways that were regulating the process of killing of Aspergillus conidia. However, it was very clear that reactive oxygen species production, ROS production, was an important effector pathway for phagolysosome killing, fusion and killing of the fungus. And in the more well-known risk factors of the disease, the genetic one, which is the chronic lymphomas disease, and the acquired one, which is the steroids, blockading ROS production was somehow associated with phagolysosome defects, inability to restrict conidia development of invasive disease. However, there is a big question, and still is there, whether ROS are just effector molecules or they are also some kind of signaling molecules that somehow regulate this physiological process of phagolysosome fusion. And a few years ago, after the discovery of TOL-like receptors, it was obvious that defect in signaling in TOL was translated to defect in phagolysosome fusion. However, the molecular pathway that was regulating this process was completely unknown, until the focus came on autophagic machinery. And in 2007, the group of Gordon Douglas Green realized that particles that contain microbial ligands and trigger TLRs recruit certain members of the autophagic machinery that regulate phagolysosome fusion and killing of the pathogens. And there is a lot of interest and a lot of discussion about this process of LC3 decoration of the phagosome and the process that this is happening, which is called LC3 associated phagocytosis. And this LC3 positive phagosome formation or LC3 or liposome formation is a very specialized immune response, which is very distinct from canonical autophagy in many terms, including the lack of double membrane, the lack of pre-initiation complex, the requirement for specific regulators like the Rubicon and the NOx and the NDPase oxidase, basically the enzyme that produces ROS, participation of certain phospholipids, especially PI3Ps, and a particular requirement for certain domains of ADG16L1 that anchor on the single membrane of the phagosome and bring the autophagic machinery, which at the end facilitates fusion of the maturing phagosome with the lysosome and results in elimination of pathogens and many other physiological processes. And this, how we call lab pathway, is not only important about host defense, let's say, and against pathogens, but also regulates a lot of physiological processes from erythrocytosis to antigen presentation to cytokine response to recycling of receptors and to anti-inflammatory, let's say, immune homeostasis and regulations of inflammation. And a lot of these mice with defects in components of the lab pathway, they tend to develop a lot of autoimmune, inflammatory, and infectious diseases. So back in 2013, Irene from our group was interested to see whether this lab pathway was activated by Aspergillus. So she infected, killed, and live conidia, and she quickly realized that only live conidia triggered this LC3 associated phagocytosis response on the phagosome. As you can appreciate here, this is a red color of LC3 decorating a phagosome of swollen conidia. So some kind of cell walls remodeling was required in order for the LC3 associated phagocytosis to be activated. And through a lot of genetic and molecular studies and biochemical approaches, she was able to understand this process. She was able to understand that this cell wall remodeling was resulting beta-glucan exposure on the surface, which was triggering DECT1 receptor signaling through the SRC kinases and ROS production, which was the master regulator of LC3 lipidation and phagolysosome killing. So immediately we realized that this induction of the lab pathway was very important for killing of the pathogen, and it was triggered by pattern recognition receptors with DECT1, the predominant fungal receptor for beta-glucan, being the master regulator of this pathway. And not only that, in the setting of diseases that are associated with aspergillosis, such as CGD, which is the congenital immunodeficiency in the subunits of the complex of the NDPase oxidase that regulates ROS production, there was profound lab blockade. But the same also happened when in patients that received steroids, and interestingly, steroids blocked upstream of ROS the activation of SYK and SRC kinases. So it was obvious that both acquired and congenital defects in the lab pathway, NOx-dependent, let's say, activation of the lab pathway, resulted in profound inability of the macrophages to kill the fungus because of failure of phagosome to fuse with the lysosome. So at that point, we thought that we understood everything, but after discussion, we realized that during swelling is not only beta-glucan that is exposed, but on the other side, there are many molecules that are removed from the surface. And we made the hypothesis that maybe some of these molecules could have an inhibitory role for the lab pathway. And we followed a genetic approach from another talented postdoc in the lab, Tonia Kumianaki, who was able to understand and identify a molecule of the fungal cell wall that inhibits actively this pathway, and this is melanin. So the fungal cell wall melanin is somehow blocked assembly of NADPase oxidase, and even in the presence of beta-glucan, when melanin was present, there was no activation of the lab pathway, and this resulted in the inability of macrophages to kill the fungus. And this was a very interesting finding, because apart from masking of pumps, it seems that these pathogenic fungi like Aspergillus, they have the ability to actively inhibit the process of phagolysosome fusion by targeting this lab pathway. And as you all know, there are other emerging fungal pathogens like the mucorales, which are also heavily melanized. So we immediately got our interest to see how these fungi are interact with the macrophages and whether the lab pathway was also playing a role in the process. And we were very surprised to identify that, as opposed to Aspergillus, which is, let's say, relatively rapidly cleared in physiological mice during infection, Mucorales conidia persisted for many days inside the macrophages of normal mice. And here you can clearly see that there are some extracellular conidia, but a majority were intracellular, sitting inside CD68 positive, CD11C positive alveolar macrophages, which were sorted, and we proved that the fungus was live. And so this intracellular persistence was not only evident in healthy mice, but also in patients with disease. And here you can appreciate a giant macrophage that has a fungal conidia inside. This is coming from a patient who has the mucormycosis. And when we looked on the mechanism of this intracellular persistence, which was very interesting, we immediately realized that the lab pathway was completely inhibited by Mucorales fungi, as opposed to Aspergillus, which was triggering lab pathway after a few hours of infection. And this also correlated with complete phagosome maturation arrest in the case of Mucorales. And if you see on the bright field, on the left part of this image, you don't have to be genius to realize that there is a lot of material which looks like melanin on the one case versus the other. And also, if you make comparative, let's say, pathology studies at different types of infection, you also realize that in Aspergillus, you have this intracellular swelling of the fungal conidia, which results in the lab activation, the killing of the fungus. However, in Rhizopus, there was no intracellular swelling. There was a persistence and a dormancy of Rhizopus conidia, which retained the same size inside the phagosome, which we proved later that was associated with surface persistence of the melanin on the fungal cell wall. And that was the case of persistent prolonged inhibition of LAP. And indeed, if you chemically or pharmacologically inhibit melanin production, you can see immediately activation of LAP pathway, phagollasome fusion, cathepsin D, positive phagosome coming, and clearance of the fungus. So that was another case of a pathogen which, instead of inducing cell wall remodeling during the process of growth, retained surface exposure of melanin in a dormant condition, and this resulted in the prolonged persistence of this fungus inside the macrophages and the ability to survive for many days, which was very interesting for us. So next question was, what is this molecular mechanism that melanin targets the LAP pathway? What is the target of melanin? What is the precise molecular interaction on that? And this process was also evaluated by Irene Kirmezi, who was able to notice by doing calcium imaging studies that melanized conidia, let me play the video here, the albino conidia were able to induce very predominant and persistent calcium fluxes during phagocytosis, whereas the melanized wild-type conidia were phagocytosed without any calcium influx. So this immediately gave us the idea to look for interaction of melanin with calcium responses and to the biochemical property of melanin pigments. And we realized that in the literature, there was a lot of association between melanin and the ability to bind metals and calcium in particular. So Irene went through a very elegant and comprehensive study to prove that during phagocytosis, all kinds of melanized conidia retain have the ability to scavenge calcium inside the phagosome, the calcium what is coming from the air. And by scavenging calcium and blocking the efflux of calcium from the inside, from the lumen of the phagosome to the cytosol, the melanin resulted in inhibition of calmodulin recruitment to the phagosome. And then Irene went to identify that calmodulin is a master regulator of LAP, because calmodulin regulates both PI3P recruitment to the phagosome and NOx activation. So by blocking calcium, melanin completely inhibited lab pathway and the killing of the fungus at the very early stages of phagocytosis. And during the process of cell wall remodeling, the removal of melanin allowed for the calcium to flux out of the phagosome to bring calmodulin and to reconstruct the liposome and to result in phagolysosome killing, fusion and killing of the fungus. In another, so it was the physiological part of the interaction of the fungi and the macrophages, and how the competition results either in killing or in persistence or in failure and invasive disease. And there was another interest in the lab by Tonia Omianaki to look on whether defects in the liposome formation, defects in the lab pathway have played some kind of important role in pathogenesis of sepsis-induced immunoparalysis, which is a well-known factor for invasive aspergillosis nowadays. So for that, Tonia had to evaluate patients in the ICU with admitted with severe sepsis, and she immediately realized that there was a group of patients, almost 1 out of 3 patients with severe sepsis, that had profound defect in LC3 positive phagosome formation during in vitro stimulation. So this lab defect was clearly associated with defective killing of Aspergillus and a profound inability to secrete IL-6, and also with a high risk for secondary infections, which is a hallmark of immunoparalysis. So it seems like lab defect is was clearly associated with defective ability to kill secondary pathogens, opportunistic pathogens, and lab, let's say, defect was a biomarker of immune dysfunction, or how we call immune paralysis in sepsis. And Tonia went further and realized and discovered the molecular mechanism, which is regulated by IL-6 signaling. And interestingly, IL-6 is one of the cytokines that are very important for the physiological cytoskeletal organization of the cells. As you can appreciate here, what happens in IL-6 knockout macrophages, they fail to have a proper microtubule network, and this is related to the inability to the important role of JAK2 downstream of IL-6 to regulate, let's say, cytoskeleton. And to make it short, Tonia identified a very important mechanism that regulates phagosome formation in lab. And now, from one part, you have the cytokine signaling coming from IL-6, which physiologically regulates cytoskeleton and endosomal trafficking that are important for phagolysosome fusion events and liposome formation. And from the other part, you have the detecting signaling pathway, which regulates ROS production and liposome formation. And hypothetically, defects and practically in experiments, defects in both arms of this pathway results in the same defect: inability of liposome formation, inability of phagolysosome fusion, and inability of the cells to clear fungi. And this is a very novel and important mechanism, because this synergistic immunodeficiency we nowadays see in a lot of patients that come with complex immune defects, like the ones that take, like say, that are in the ICU and take tocilizumab for COVID-19, or take JAK inhibitors for COVID-19, and in the top of that, they take steroids, because simply you have defects in both the trafficking and the signaling pathway that regulates lab, and this synergistic inhibition of lab pathway creates a background of profound immunodeficiency, which we can hypothesize that will lead to susceptibility to fungal disease. And indeed, to our surprise, after publishing this study, there was a study that evaluated risk factors for kappa, COVID-associated pulmonary aspergillosis. And of course, as you all know, these patients are already on steroids. But on the top of that, it seems that IL-6 inhibitor, tocilizumab, was an independent risk factor for invasive aspergillosis, meaning that the work of Tonia identifying the mechanism and identifying the defect of this pathway was a very important contribution to understand how the disease is happening, and to identify a biomarker of this susceptibility process. And to summarize, so far the studies that have discussed, it seems that phagosome defects projecting lab defects result in the inability of macrophages to inhibit fungal growth. This is the first line of host defense, and I think this is well appreciated. And these defects are associated with any type of disease that we have that has been linked to aspergillosis so far, including congenital syndromes, steroid treatment, targeted therapies, and sepsis. So that was an interesting part of our work. And of course, we wanted to explore whether this is this lab pathway is an evolution conserved mechanism. So in the Drosophila model that we are studying right now, it seems that lab is very well preserved, and defects in the genes regulating NOx and other liposome components result in inability of Drosophila hemocytes to restrict the fungus, which is very reassuring, meaning that this is an ancient immune mechanism in the phagocytes, at least in the macrophages, to encounter this opportunistic fungi metabolism to target the metabolite on the phagosome and block the ability of a pathogen to invade when the melanin is blocking phagosome fusion. And here I will stop, and I will thank, first of all, Irene and Tonya Irene, which have been doing all this work for the last 10 years, and the newer members, Vasilis and Adonis, and the new members of the lab, which will come in another photo, funding especially from the ERC, and the important contribution of collaborators, especially Jean-Paul Adje, Agostino Carvalho, Michalis Ionakis, Frank van der Wirdok, and many others, and Ashraf Ibrahim for sure. And thank you all for your attention.
Thank you very much. So just to remind our audience, we're going to go straight into the second talk, but if you please start entering your questions now at the Q&A, and remember to put the initials of the speaker before your question, and we'll come back at the end of the second talk. So our second talk now will be from Professor Anuradha Chowdhary, who is Professor of Medical Mycology and is also in charge of the National Reference Laboratory of Antimicrobial Resistance in Fungal Pathogens at the University of Delhi. So she received her medical training from the University of Delhi and later a PhD in the Netherlands, followed by postdoc in the USA. And Dr. Chowdhary's research interests include molecular ecology and population genetics of pathogenic fungi and antifungal drug resistance. And she's currently working on Candida auris, and that's what she's going to talk to us about today. So when you're ready, you can share your slides.
So thank you very much for the introduction and for this opportunity. So today I'm going to talk about our work, which we have focused during last 3-4 years.
If I could interrupt briefly, you need to switch into presenter mode.
Yeah, sorry.
Thank you.
Thank you. So I'm going to talk about Candida auris work, which our lab is pursuing for last 3-4 years, where we are mainly looking after the distribution of Candida auris in the natural environment outside the hospital settings. And also we're trying to understand its transmission, its emergence and transmission. So Candida auris is one of the global pathogen listed first in 2019 by antibiotic resistance threat report by CDC, and recently by in the fungal priority pathogen list. It has been listed under the critical group because of its antifungal resistance in this pathogen, and also its ability to get transferred from one patient due to its ability for its nosocomial transmission in ICU settings. And why this is it still remains important issue, not only in the North America, but also in the several parts of the world. But North America has especially United States has a very important system of noticing and recording these cases. And this is one of the recent report from CDC, which shows that how the worsening spread of Candida auris is occurring in the United States, where the CDC shows that 44% increase in Candida auris cases has occurred from 2019, and the percentage increase is now 95% into 2021. And also what is important is that the rate of resistance, especially the kinocandin resistance in Candida auris, has increased 3 times than in each of the previous 2 years. So C. auris remains an important pathogen. And although the retrospective analysis of large number of isolates, large collection of isolates have shown that this emergence is actually recent, because isolates dating back from 1996 has not been observed. So this is just one of the case where I wanted to highlight that probably Candida auris existed in India before even it was labeled as a species, which was in 2009. So this is one of the case report from France, which showed that one of the patient who had visited India in 2007 and was admitted in one of the Delhi hospitals acquired Candida auris. And when the patient was shifted back to France, the isolate which was recovered from liver abscess was not able to identify due to non-availability of ITS sequences at that time. And retrospectively, this species was identified by the National Reference Lab, and it was Candida auris belonging to clade 1 from South Asian clade. And this work prompted us to also find out in our culture collection to see if the isolates were there before the early outbreaks when we started working in 2012. And we saw that few of our early outbreak isolates, when we estimated the diverse emergence of Candida auris, we found that few of our isolates had the divergence time probably in 2007, and those two isolates of candidemia was quite similar, very related to the French isolates, suggesting that although we know that India has the Indian subcontinent Candida auris is endemic, but probably it existed even before the species was labeled. So, so far the Candida auris remained, I mean, during the time of COVID pandemic, several reports emerged later, which showed that the transmission of Candida auris in ICUs was enhanced, was increased during that time. And this is one of the very early study during the pandemic, where we showed that during the April and July of early period of pandemic, we showed that 2/3 of the cases of candidemia, which patients had and who were severely admitted in ICUs, but had this breach of, they had this respiratory failure. And in those patients, we saw that 2/3 of the cases were due to Candida auris candidemia. And the remaining risk factors were similar as seen in other ICU patients. But what was significant at that time was that 40 to 40% of the isolates which we had were resistant to two classes of antifungals, and the most commonly used antifungals like azoles and amphotericin B, about 30% of our isolates were resistant to both the classes of antifungals. And also the case fatality rate was high. And this prompted us to find out whether the incidence of transmission of Candida auris has increased during the time of COVID-19. So we did a larger cohort of more than 2,000 COVID-19 patients in 2 ICUs. We found that incidence of 1.4% over candidemia in patients who had a prolonged admission in ICU for more than 24 days. And we also saw that there was a 2-fold increase in the incidence of candidemia in COVID-19 patients. So these studies basically highlight that Candida auris always remains significant in the hospital settings. And in this case control study, we also observed that ICU stay was one of the important risk factor, and also the IL-6 inhibitor, and also the raised ferritin levels were important contributors for the development of candidemia. So basically now, so far, I mean, I have convinced that it is an important pathogen inside the hospital settings, and because it caused large outbreaks. But now I'm going to talk about its transmission in the natural environment. And as I said, that probably it emerged in the 3 continents simultaneously. And one of the hypotheses which was put forward was that global warming probably resulted the emergence of the C. auris as human pathogen. And as we know that Candida auris can survive at higher temperature at 42 degrees Celsius and also can tolerate the hypersaline environment. So this hypothesis was tested during the time of COVID-19, 2019 and 20. We used that time to collect the samples from the marine environment, from Indian Ocean. And these are the areas which we chose, which are restricted for human activities. And we collaborated with the Marine University, and we collected samples from Andaman isolates, which had a varied topography. So we collected from salt marsh, from sandy grooves, from the beaches, and these 8 sites which we collected the sample for the seawater as well as the sediment, we found that 2 sites where we could isolate Candida auris. And what was important from these 2 sites was that one site which was representing the beach site where the human activities was there, where there we had large number of, more number of Candida auris as compared to the site where there was no human activity. There we had only 2 isolates which we could recover, and that was from a salt marsh. So these 2 isolates now also differed in the susceptibility pattern. So basically the isolates from the beach site had all the parameters of antifungal resistant genes, and they were all resistant to multiple classes of antifungals, including fluconazole. Whereas this susceptible isolate, which was from a salt marsh with no human activity, that was found to be susceptible, low MICs for fluconazole. And also the isolate which had a low MIC for fluconazole, which was from the from the restricted human activity, was growing slowly at 37 as well as at 42 degrees Celsius, and which suggested that probably the acclimatization of at higher temperature or growth patterns at higher temperature occur slowly, and we presume that this isolate probably is the susceptible isolate which is not exposed to the human activities. And also the genomic analysis of the isolates showed that they were actually distinct from, they were in the clade 1, but largely they were clade 1. But the susceptible isolate was not related to many of the clinical strains from India, whereas the beach isolates were more related to the clinical strains from the Indian clinical strains. And this is our recent data of the Andaman isolate, susceptible isolate versus resistant isolate. And here, when we look at the resistant isolate differential expression analysis, we found that ergosterol biosynthesis genes were upregulated in resistant isolate as compared to the susceptible isolate. And also the transporters, these we validated, and all these transporters were upregulated in the resistant isolate. And also we observed that copy number variation in the susceptible isolate as compared to the resistant. A single copy number of ERG11 was seen in fluconazole susceptible isolates. So probably all this hints to the presence of that what is driving the resistance in the environmental isolate is probably the continued pressure of antifungals which are driving this changes. And and this is the second study which came after the Andaman isolate, where they also, from Colombia, where they also found that the samples which they collected in 2018 from several marine and estuary bodies of water across Colombia, one of the sample was positive, and that has a mixture water from the fresh water as well as from the sea water mixing was occurring. And so if you can see at the low salinity also, the Candida auris, which was isolated, had this kind of presence of these parameters in the water. So basically, both these studies suggest that Candida auris can persist in a natural environment and water. And here, I like to show this study where this study has shown the applicability of the presence of Candida auris in water, and they have done the wastewater surveillance for C. auris and to detect the outbreaks which were occurring in the metropolitan area in Nevada, where multiple outbreaks were occurring in several healthcare facilities. And here they used qPCR for targeting the ITS2 region of Candida auris. And they found that 79% of the water samples were positive. Wastewater surveillance, both primary influent and effluent, was positive for auris. And what was more important, that 90% was samples from the sewer sheds, which were close to the hospital care settings, were positive for Candida auris. And the same group of workers have published a second study where they have isolated in culture the Candida auris, and to see whether the strains which were in the water were related to the to the strains which were found in the human cases. And of course, they found that clade 3 was responsible for the majority of the outbreaks in that healthcare facilities, and the same clade was isolated in water. So suggesting that this technique can be used for surveillance of Candida auris and can be an important public health tool. And then the second part of our study focused on how this resistance development is occurring in the environment samples. So here we know that the fungicides, which have a similar structure as many of the azoles which are used in our human practices, so that drives antifungal resistance in Aspergillus fumigators. So what we did was we identified several niches, and we also focused on fruits which are stored because they are primarily sprayed by a lot of fungicides. And here specifically apples, which has a long shelf life and which are stored for a longer period. And here we screened about 62 apples for over a 1 year period, and we saw that those stored apples which had a high concentration of diazole, triazole, and other fungicides, all these classes of fungicides which was present in the stored fruits, apples. Here on the surfaces we found C. auris. On 6 of the apple surfaces we could found C. auris, whereas the seasonal apples did not have any of these fungicides, and also even the organic apples which had no fungicides, none of the auris isolate was present on the surface. So and the DMI fungicides, when we detected the fungicides in the apples, we also wanted to do the susceptibility testing for these strains. And what we found that tebuconazole and fluconazole, for these two fungicides, the MICs of these strains which are obtained from the fruit samples had very high MICs for both these fungicides, suggesting that the continued pressure of fungicides probably not only result in the high MICs of fungicides, but also cross resistance to fluconazole. And also interesting was that a lot of strains on one surface apple, we had 2 or 3 genetically distinct strains. And also we found that the apple surfaces which were having these genetically distinct strains, when we analyzed this population with clade 1 strains from India, we observed that they were not only related to the clade 1 strains from human patients, but what was interesting was that few of these strains was also related to the strains outside India. So one of the strains which was quite similar to the apple strains was from a patient from France. And this patient had a history of travel to India. And also 2 of the strains from the Canada were closely related to the fruit strains. And these patients also had, one of the patients had a history of hospitalization in India. So probably these findings suggest that the transmission of Candida auris is occurring through various sources, and probably fruits can be one of the sources which contribute to the transmission of Candida auris. And the surfaces of apples, when these are the number of species of yeast which we isolated from the surfaces of apples. And if you look, not only Candida auris, but also very important species of Candidemia like Paropsilosis, which is very common not only in Indian subcontinent, but also in other part of the world, like Candida krusei, Candida tropicalis, all were found on the surfaces of the yeast. And also was also significant that first, when we found out Lodromyces elongisporus, we never had seen a case of Lodromyces elongisporus till last year in 10 years of our practice, and we did not have any of the clinical strains of Lodromyces elongisporus. But recently, we had to investigate an outbreak of fungemia due to Lodromyces elongisporus in our neonatal intensive care unit. And I am talking about this because it has a very interesting connection with these apple strains. And these 10 neonates had this a period of about 6 months. We had 10 neonates who had candidemia due to fungemia due to Lodromyces elongisporus. And if you look at the risk factors, they were all preterm or low birth weight babies. And they were admitted in one of the single neonatal intensive care unit. And when we did the susceptibility of these isolates, we found that clinical bloodstream isolates were susceptible to fluconazole. In addition, we also isolated Lodromyces longisporus from the neonate warmer and from the temperature panel, as well as from the railing of one of the neonatal warmer. And we thought this environment, inanimate environment strains are probably persisting, and probably they are causing the continued transmission of these fungemia outbreaks. And also, we included in the whole genome sequencing the strains which we obtained from our auris studies, these 13 strains of Lodromyces longisporus. And when you look at the susceptibility pattern, you'll see that the strains which were obtained from the surfaces of apples, which had a high MICs which had a high content of fungicides, had high MICs of fluconazole here as compared to the clinical strains. And also, when we did the genome sequencing, we found that the clustering of cases of clinical patient from the neonates, as well as the inanimate environment from the patient warmer. And interestingly, 2 of the fruit strains also had a high genetic relatedness with the clinical as well as environmental strains. So suggesting that there's a mixing which is happening, the transmission is happening inside the environment, also outside the hospital environment. And we were not able to contain the outbreak in the initial 6 months, despite reviewing our infection practices, control practices, despite using a lot of disinfectants for cleaning. So we also did the susceptibility of all these strains with the disinfectant, high grade and low grade disinfectants which we use, and we found that hypochlorite, which is a common disinfectant used for cleaning, we found that several of our strains, for clinical strains, 50% of the clinical strains, as well as both the inanimate environmental strains, had high MICs for sodium hypochlorite, more than 1% recommended concentration, which is used in the clinical practice. And also, one of the fruit stain which had fell in this clinical cluster had high MIC of the hypochlorite. So when we tested in the presence of hypochlorite, we grew this Lodromyces, we found that these strains, inanimate environment strain, were growing in presence of sodium hypochlorite, even at the 2 hours and 8 hours. And also, we also did the FACS analysis, and we found Lodromyces longisporus is a diploid yeast, but the inanimate environment strain from the warmer, both these strains had aneuploid and was aneuploid, and which also suggested that, and also the strain had a loss of heterozygosity at scaffold 2, and this all suggests that probably stressors in the clinical settings like disinfectants or antifungals driving changes at the genetic level. And this study I wanted to highlight because this is one of the attempts from these investigators to find out using the DNA meta-barcoding data. They have screened the NCBI database for Candida auris specific ITS sequences. And although this study shows a presence of ITS, Candida specific auris ITS reads in different animals, like they were able to find out this trace sequence of Candida auris ITS in the ear skin of dog, although the reads are very less, like 0.01%, 12 reads out of 81,000 reads of ITS. And also that they could find out in the Nuet from UK study, the skin surfaces of the Nuets had traces of Candida auris. Basically, this study hinted that a lot of other animals are also probably involved in as an intermediate host or as transmission of Candida auris, which we are not aware. But why I'm showing this study, that this prompted us to look for Candida auris in dogs, and this is our unpublished data. But I'm sharing here because this is one of the recent work which we have done, and this study prompted us to look for Candida auris in a large cohort of dogs in Delhi. So basically, we screened about 87 dogs, and we made 3 cohorts of this. And the one bigger cohort came from a big hospital from Delhi, where they have diseased dogs, chronic skin infections. And in the chronic skin infection group, we had a small group where the dogs had a confirmed otitis externa. So these were the 8 dogs. And these dogs, we screened for the presence of Candida auris from the skin samples, from the lesions, as well as from the ear samples. In addition, with the otitis externa group, we also did the ITS amplicon sequencing for fungal ITS for looking for Candida auris specifically. And what we observed was really exciting, that in 2 of the dogs, we had isolation of Candida auris from the ear canal. And one of the group was otitis externa group. And also in this group where the Candida auris was from ear infection, this auris was present along with the bacteria, with Acinetobacter baumannii and Pseudomonas intermedius. So it's kind of a mixed infection. And Candida auris persisted in this D1 dog for almost a month. And we also had a second isolation after a month, and the strains were highly related. And in the other group, where from the ear canal, we had isolated Candida auris, but the patient did not have the otitis externa or any ear infection. And in addition, we found high reads of ITS in 3 of the specimens, 2 from the ear swabs and 1 from the skin swab of the dog. So overall, we could find that the association of Candida auris in the ear canal of the dogs, but a lot still has to be done. But what was interesting was that the reads which were high, compared to the previous study from Wheeler and Meyer, where it was in 0.001%. Here we found 0.12% of the auris reads by metagenomics in the dog's ear, which had isolation of Candida auris in culture, whereas we had a low reads in the dogs where we were not able to isolate in the culture. And in addition, when we did these are the heat maps, which shows large number of flora, I mean, the genus of Candida and other molds. And also, what is interesting that we saw, not only the relatedness of these strains, the dog strains, with the clinical strains from India. The first D1 dog, which had 1 month infection, which persisted, those strains were quite similar to the strains which were obtained from the Candidemia cases from Indian subcontinent. But the second dog, which we recently did, was positive for Candida auris. That strain was quite similar to the strain from a Pakistani reference strain. And we're not very closely related to the strains from Candidemia from India or from the ear swabs of patients, but with the Pakistani clade. So overall, we're still figuring out, but overall, I mean, our work just emphasized that C. auris has ways and means to persist in the environment, as well as probably several animal species which we have not yet, which a lot of things has to be done, which have not been discovered. But these are these areas where it require the studies are required, focus studies are required. And this is what we presume that is happening at the level of in the environment, in the concept of One Health, that not only the wetlands, but how it is transmitted to humans. There are several reservoirs and factors which are new and which are still need to be discovered. But probably, I don't know whether it's a concept of One Health or not, but definitely the environment has a very important role. And Candida auris has several ways where it can introduce itself in the environment and can have a cycle which is very important, the transmission cycle which is occurring outside the hospital settings. So with this, I'm thankful again to all of you for listening, and to the entire group of Patel Chest in my lab, all the students for last 3 years who have been working in identifying the several issues of Candida auris. And thank you very much. And I hope I'm in time.
Yes, perfect time. Thank you. Okay, so can I remind our audience to please submit their questions under the Q&A, and don't forget to put the initials of the speaker in front of the question. Okay, so I'm going to start with a question that to Georgia that you've already answered, but just so that the rest of the audience can hear. There was a question as to whether the mechanism against aspergillosis works in cancer patients.
You want me to repeat that?
Yes, because the audience can't see your answer.
As you all know, not all cancer patients develop invasive aspergillosis, not even the high risk ones. So in a solid tumor patient, the risk is in general low. However, if somebody is taking, for example, corticosteroids or let's say ibrutinib or other type of small molecule kinase inhibitors, or the ones that I described in my talk, like JAK inhibitors, there is an increase in the risk. And there is a good possibility that the LAP is inhibited in this process, but we don't know for every patient if LAP is inhibited.
Okay, thanks. For Anuradha, there's a very a question that's addressing all of your talk, really. How do you think that Candida auris is acquired? So do you think it's part of the human microbiota, or is it coming from the environment?
So yes, it is an important part of the human microbiota because the colonization occurs on the skin surfaces, on the skin of the ear. It has been isolated from the urine of asymptomatic patients. It has been isolated from the fecal flora. So yes, probably it is there on the human skin, and it doesn't cause infection until unless the skin is breached, or the immunosuppression is there. So environmental sources outside hospital, we are just struggling. We're just identifying. But what is already proven is that its presence on the human skin as well as in the mucosa, as well as it is there in the humans.
Yeah, and a point that's been raised too is when you tested the dogs, did you test the owners or the people that were closely associated with them?
Yes, we did. I did not show that because it's at the part of the paper. We also did the ICU healthcare workers who were handling. And we also did the microbiome analysis of one of the healthcare worker because it had a heavy load of Candida parapsilosis and Candida tropicalis. But none of the workers' hand or the floors had Candida auris, no. So these are the dogs, major cohort of the dogs were in the intensive care units, which were either chronic skin diseases, and they were stray dogs. So the group which did not have Candida auris, the group third, 35 dogs, which I did not discuss because they were healthy dogs, but they came for GI complications or other diseases, mild diseases. And they were outpatient dogs. So none of the auris isolate was able to isolate from the that cohort.
Okay, we have a lot of questions for you.
I was thinking to share this because, very interesting thing happened when we submitted this to one journal. So at that time, the study was not complete. It was just 1 or 2 isolations from, small. But then we did last 3 months extensive study, all the microbiome analysis, more thorough. So then we had this clear picture. But then I thought I have to put it in front of all of them.
And we've been asked for a clarification of how you identify Candida auris. So is it always ITS sequencing, or what do you do?
Yeah. So basically, if you're talking about the isolation, once we have in the culture, we have a culture plate. So we use MycoTalks. We have a huge database, our own database in Bruker system. And then we also confirm by ITS sequencing because, we still feel that a lot of strains require, because sometimes the MycoTalks gives a low score. So yes, we always do ITS sequencing from the isolates. But for isolation from the water sources, from the dogs, these are isolation, and we never did the qPCR directly from any of the clinical samples.
So when you take them from the apples, you culture them?
Yes, we cultured the apple surfaces, and we used a specific media, enrichment broth media, which has a high sodium chloride and dulcitol, which is recommended for isolation from the patients for colonization.
So that's enriching for Candida auris? It is.
Because of saline and because of dulcitol, yes.
Okay. And one thing I was wondering also is, why did you choose those sites in the Andaman Islands, and why marine sites? Why is everyone looking at marine sites?
Because of the hypothesis put forward by Dr. Kasadawal. I mean, I was really fascinated when I read that paper, and we had enough time during the time of COVID pandemic. And that hypothesis was like probably Dimitrios and Kasadawal group gave this hypothesis that probably Candida auris, because of its tolerance to the saline, and also thermotolerance, it probably is associated with wetlands, and also Metchinovi complex, where the Candida auris belongs to this Metchinovi complex. Several other species like Lusitania and others have been isolated from seawater or from marine, animals. So, because it belongs to that clade, so it was speculated that probably it has some relation with the marine bodies, and which we thought it existed. It has. So yes, that is why we did it.
Okay, right there. So can we get? I would like to encourage people to ask Giorgio questions. So can you get typing, and hopefully while you're doing that, Anuradha, are there any screening strategies for Candida auris in Indian hospitals now? You think it's an important enough pathogen to screen for it?
Sorry, I did not.
So the questioner is wondering, do you screen regularly in Indian hospitals?
Yeah. I mean, at least in our hospital, we do it. And I wanted to present that study, but then I thought it's not related to the natural environment. One of the very interesting, we screen because we have chronic patients. I've come from a chest institute. We have chronic patients who keep on coming and going because of asthmatic attacks and other respiratory diseases. So when we did the colonization study for the first time, we saw that about 7 to 10% already before coming, because of the repeated admission, had Candida auris on their skin. And within a week when they're admitted, they become colonized by Candida auris. And not only that, by genome sequencing, we found that different genetic strains are there in the nose and the ear, and also the similar strains they shed in the environment. And within the 7 days, this close environment becomes positive. So yes, we screen, but since ours is more of a chest institution, so probably we do not have seen a major candidemia occurring after that. But several studies globally has shown that there is 20-25% chances once the colonization occurs to have candidemia. Okay.
And we have a question for Georgios. How many melanin biosynthesis pathways do rhizopus have?
No, it's a good question because it doesn't have a clear answer. We don't know the genes for biosynthesis of melanin in rhizopus. We do know that it's not a DHL melanin; it's a dopamelanin, and there is no mutant available yet.
Okay, so that's going to be
genetics are difficult in rhizopus.
Yes, that's what I was imagining. So are you going to have to develop systems to take out all the pathways? Probably. Yeah. Okay. Do we have any more questions? Actually, there's one I'd like to ask about Candida auris, about the drug resistance. So when it's described as resistant, how is that resistance compared to related species? So things like Clavispora, Lusitania, or other species in the same clade.
Yeah. So the species in the same clade are also, that's a antifungal resistant clade actually. Lusitania is resistant to amphotericin B, yeah. And but when we analyze a huge collection of isolates, we have like an Andaman isolate, we have one isolate which had low MICs. So we also had susceptible isolates because we do not have still the cutoffs for these are the CDC proposed cutoffs, right? But we do not have CLSI cutoffs still for Candida auris. So we still have about 10% isolates in our collection, which show low MICs for fluconazole. When I say low MICs, like 4, 8, 16. So that also hints that probably it's not inherent resistance. It has developed over the, continued pressure. So, and that was also the finding in one of the susceptible isolate here.
Okay. And do you think the method of storing apples, do you think that's going to vary from country to country? Would you have different levels of auris in different places?
Yes, of course. I mean, which and fungicides are used where, how many, and it varies. Yeah.
Yeah. Okay. I think we have asked all the questions unless I don't see anything.
Yeah, one of the question which I now see, Denalila, he has asked me whether you screened the owners of dog, but I want to clarify here the dogs were stray dogs, many of them, right?
Yeah.
Like 80% of the dogs. So no, but yes, I don't think that dogs which are from the community, which pet dogs, because although we did 35, but they did not have, and they have a very different skin flora as compared to the diseased dogs. I did not wanted to show the photograph because it was like too much. And then I thought, so it's a very preliminary study. I mean, I'm not saying that dogs or the other reservoir or they're helping in transmission. No, I'm not saying that. I'm just saying that yes, it has an association similar to the like humans, like ear. I mean, it has something with that. Also on the skin, similar as humans get colonized, but in a very small fraction. And that too coming from a place where Candida auris is almost there in India, right? So this varies in different places.
Okay. Okay. I think we have asked and answered all of the questions. If there are any more that come in, we'll send them to you later to look at. And thank you very much. That was 2 fascinating talks.
Thank you.
We'll
Thank you.
hopefully see everyone back in a month. Okay. Thank you. Bye.